All right, in this last section, we want to go over chromosomal inheritance. So normally an individual is going to receive 22 pairs of autosomes and 2 sex chromosomes. Each pair of autosomes is going to carry alleles for particular traits. Those alleles could be different. So like I said earlier, you might contain one allele for brown hair from your mom and one allele for light hair from your dad. We'll talk quite a bit about this a little bit later on, but those alleles can be different. Or they may not. You might inherit brown hair from both your mom and your dad. Alleles can be the same, can be different. We'll talk about this a little bit later on. What we want to focus on specifically in this section is changes in the chromosomal number. Some individuals may be born with too many or too few autosomes or sex chromosomes. We can actually see changes in this chromosomal number occur in both autosomes and sex chromosomes. This is going to be caused by a process known as nondisjunction. Nondisjunction during either meiosis or meiosis 1 or meiosis 2 means that we're going to have a failure to separate. We made a mistake during our seatbelt check. Something wasn't locked down properly, and we have a failure to separate either the homologous pairs or the sister chromosomes. A trisomy is what occurs when we have only one type of chromosome, or when we have one type of chromosome present in 3 copies. So when you end up with an extra copy of a chromosome, this is said to be a trisomy. When you end up one short of a chromosome, so you have too few chromosomes, this is said to be a monosomy. Tri meaning 3, mono meaning 1. There's only one type of chromosome that's going to be present. So we might see an egg with 22 chromosomes instead of 23 chromosomes. Could then be fertilized with a normal sperm. We're going to end up with an individual with 45 chromosomes instead of 46. Here is how that would occur. So if we have nondisjunction in meiosis 1, so we have originally one pair of chromosomes. If they fail to separate, we failed our seatbelt check. We didn't separate. We didn't make sure that the seatbelt was tightened down properly. Both of those chromosomes are going to end up in one cell, and another cell is going to end up without any chromosomes at all. What we have over here is, if those cells were then to undergo meiosis 2 without any further problems, we're going to still see that although we separated those sister chromosomes, we still have 2 copies of it. We have this sister chromatid here and this sister chromatid over here. We have this sister chromatid here and this sister chromatid here. So we've still ended up with additional chromosomes. If we then are fertilized by a sperm cell that is carrying 1 chromosome, we're going to end up with 3 chromosomes over here. 3 chromosomes over here and only 1 chromosome over here. Over here we have a trisomy, over here we have a monosomy. We have 3 chromosomes, here we have only 1 chromosome. This can also occur during, several stages. So here we have oogenesis. If we have a nondisjunction during oogenesis, we can end up with, here we have a normal meiosis 1. We have our homologous pair of chromosomes. They separated correctly. This cell may go on to separate correctly or incorrectly. So if we lock down our seatbelts, we do our seatbelt checks, we could end up with meiosis 2. We could end up with 2 well-divided sister chromatids. So here we separated those sister chromatids. They're then going to be fertilized, and we're going to end up with a correct diploid number of cells. We have 1 of each copy, 1 maternal chromatid, 1 paternal chromatid. If we have a nondisjunction occurring in meiosis 2, we're going to see that although we separated the homologous pairs, these sister chromatids might not separate. And in that case, we're going to end up with 2 identical sister chromatids over here. We're going to end up, once we fertilize, we're going to end up with a trisomy. And since there was no chromosomes contributed by the mother over here, but we have 1 chromosome contributed by the father, we're going to end up with a monosomy over here. So this is the consequences of nondisjunction. If we do not undergo that correct seatbelt check, if we make a mistake at the seatbelt check, we could end up with either too many or too few chromosomes in our final stages once we fertilize that egg cell. Now, there are a lot of potential consequences for this, so that's what we're going to spend the rest of this chapter looking at, is what happens when we have changes in either chromosome number or chromosome structure. So anytime we have an abnormal number of autosomes causes a developmental abnormality. Monosomy of all but the X chromosome is going to be fatal. Trisomies are usually fatal, but there are just a few exceptions. And by fatal, I mean that very few are going to, almost all trisomies are going to result in an autoabortion or a miscarriage. Except for with, there are just a few trisomies that can survive. Trisomy 21, so 3 copies of the 21st chromosome. Trisomy 21 is the only trisomy that does survive after birth. It's the only one that has a chance of survival beyond early childhood. The chances of survival are greater if the monosomy and trisomy involves only the sex chromosome. In normal XX females, only one of those X chromosomes is going to be useful. One of those X chromosomes will be called a bar body, which will be eventually inactivated. So female cells are going to function with just one X chromosome anyway, just like male chromosomes do. Males are going to function with one X chromosome and one Y chromosome. This is the most likely reason that a zygote that has only one X chromosome can survive. So if you do end up with a monosomy of the X chromosome, you'll result in a female with only one X chromosome, they are very likely to survive. There are going to be some consequences, but they will survive into adulthood. Trisomies or monosomies of the sex chromosome. All extra X chromosomes are going to become bar bodies. So there is the potential for a poly-X female, a female who has multiple X chromosomes, an XXX female. XXY males are also survivable because those extra X chromosomes are going to simply be deactivated. An extra Y chromosome is called Jacob syndrome. So an XYY individual is survivable because the Y chromosome only carries a few genes, so having additional copies of those genes is typically not fatal. This could occur from the nondisjunction during meiosis II of spermatogenesis. In the examples we gave, we only showed nondisjunction occurring during oogenesis, but it can occur during spermatogenesis just like it would during oogenesis. As we mentioned previously, Down syndrome, trisomy 21, it's going to be the most common autosomal trisomy. It's really the only one that can survive. This is when we have 3 copies of chromosome 21. Women over 40 are more likely to have more likely to undergo that non-disjunction and are more likely to see 3 copies of that chromosome 21. There are a couple of characteristics associated with chromosome 21. This is due to the extra copy of the GERT gene. So it's a particular gene located on chromosome 21 that can result in a shorter stature, eyelid fold, flatter face, shorter fingers, a wide gap between the first and second toes, a large fissured tongue, round head, palm crease, intellectual disabilities. All of these can be associated with Down syndrome. So here is that chromosome 21. If we do have an additional copy of that chromosome 21, there's a particular GART gene located right around here that is thought to be responsible for those additional signs and symptoms. Turner syndrome, as we mentioned earlier, this can occur, this is basically a monosomy of the sex chromosome. This can be caused by non-disjunction during either oogenesis or spermatogenesis, can result in gametes that have too many or too few sex chromosomes. Syndrome is just a group of symptoms that always occur together. The presence of a Y chromosome, not the number of X chromosomes, determines maleness. If there is a Y chromosome, the individual will be genetically male. If there is, so it's about having the Y chromosome present. So if there's no Y chromosome present, the individual will be female. The reason for this is that on the Y chromosome, there is an SRY gene that produces a hormone called testy determining factor. This is going to be important in the development of male sex organs. If that gene is not present, male sex organs will not be produced. And without those male sex organs, we will not see the development of female characteristics or femaleness. So like I said, Turner syndrome, that X naught syndrome, that 0 there just indicates that there is nothing there. So it's X naught or X nothing. So in this case, the individual will be female. They'll only have one X chromosome. They tend to be shorter with a broad chest. The ovaries, uterine tubes, and uterus tend to be very small and underdeveloped. They typically do not undergo puberty or menstruate. Breasts tend to not develop. However, the individual will be of normal intelligence and can lead a fairly normal life if they receive hormone supplements to help compensate for those missing hormones. Klinefelter syndrome would be poly-X male. These individuals are going to be male. They'll have 2 X chromosomes and 1 Y chromosome. The symptoms tend to be pretty subtle. Only about 25% of males who have Klinefelter syndrome are even diagnosed. They are going to, however, require assistance They are going to require fertility treatments. They're going to require some sort of assistance in order to father children. And typically they can be helped by receiving testosterone supplements at the beginning of puberty. There are also poly-X females. Poly-X females are when we have more than 2 X chromosomes and additional extra bar bodies. There tends to not be too many distinctive changes in appearance, aside from these individuals tend to be a little bit taller and thin. They may have delayed motor and language development, but no intellectual disabilities. Children will usually have a normal karyotype. They aren't going to typically pass on that poly-X syndrome to their offspring. Females with more than 3 X chromosomes are going to be exceptionally rare. Jacob syndrome. Jacob syndrome can only result from a nondisjunction during spermatogenesis. This is where we're going to have an additional Y chromosomes. Individuals who have this genotype are going to tend to be very tall, have persistent acne, and tend to have speech and reading problems. But they tend to be fertile, do tend to produce normal, healthy offspring. All right, and that's our Jacob syndrome. The next section of our the next section that we're going to focus on is going to be changes in chromosome structure. So we've talked about changes in chromosome number. I am going to ask on our study guide that you review these changes in structure, or changes in chromosome number and structure, and that you pick one. You pick one change in chromosome structure or number and become very familiar with it. Know what causes it. Know what is the change. Are there additional X chromosomes, additional Y chromosomes? What are some of the signs, symptoms, and characteristics of individuals who have that additional or not additional, but what's the term for not additional? Anyway, monosomies or trisomies. So make sure that you just review individuals who have too many or too few chromosomes and the signs and symptoms that are associated with that particular genetic variability. The next section, like I said, is going to focus on changes in chromosome structure, so we will have a separate video for that.